Okay, so before we go any further with the discussion of photosynthesis, let's kind of go over some of the similarities between mitochondria and chloroplasts. And remember this concept of endosymbiosis. So you have this chloroplast that's sitting here. It has two membranes, an inner membrane and an outer membrane. And if you remember correctly, the mitochondria has that same situation. You have an inner and outer membrane. Here they sit. And the chloroplast also has that inner and outer membrane. A bacteria only has the only has one membrane, but sometimes they have cell walls. So that's an interesting difference between the three. But what the reason, the way we can explain these two organelles that exist inside plants, both of these exist in plants, and in animals only the mitochondria. When we algae, you're not going to get single-celled algae or photosynthetic bacteria. You're not going to get chloroplasts at all, but you're going to have the components of the chloroplast embedded in the membrane of the bacteria, so that it does its photosynthesis without the need of all these membranous organelles. Remembering that a bacteria, one of the definitions of a bacteria is it doesn't have any organelles that require membranes. So the only, things like ribosomes it has special ribosomes that are different from eukaryotic ones. They're a different size, but it has no organelles that have membranes. So they don't even have a nucleus, for instance. They have this little kind of nucleoid space, a little place where just the DNA is kind of raveled, is just sitting in there. Remembering that DNA is circular versus eukaryotic DNA is linear. Okay, all that kind of review of what we covered just a few weeks ago. So when we talk about mitochondria versus chloroplasts, what they do is very similar. And you remember the lab that we did with the batteries and the salt acid. You'll remember that the electrons moving through the fluid, through a conductor, actually the movement of electrons actually stimulates various chemical reactions that allow energy from the electron to be placed into that high energy molecule that we call ATP. Now, but before we go on to discuss how that works in a chloroplast, try to remember how it works with the mitochondria before you go on. But I'd like to remind you about this idea of endosymbiosis. We're looking at these are again a 5-step process of what we're talking about when we talk about endosymbiosis. You have initially millions of years ago, before there were multicellular organisms, there were bacteria in the oceans. One could do photosynthesis, cellular respiration, or maybe it could do photosynthesis. So one had these proteins embedded in its membrane. So this inner membrane would have been the membrane of the original bacteria that was absorbed by or engulfed by that second bacteria millions of years ago. And the same thing it would be true of the mitochondria. So the inner membrane would be the membrane of the original bacteria that could do cellular respiration really well, and in the case of the chloroplast could do photosynthesis really well. So some bacteria, some mother of all bacteria back in the day, ate the mitochondria, the cell with the mitochondria, but it didn't digest it. It engulfed it. Remember, phagocytosis, the way it works is that the cell uses its membrane. See how it's pinching around it? So what would happen is that this membrane would pinch off. So what would happen is that this thing that was engulfed would have a membrane around it that came from the bacteria that swallowed it. So this membrane, this secondary membrane, that would be around it, around the membrane that this particular cell had originally. So remember, this cell had its own membrane. So that membrane would be inside the second membrane, theoretically, if it was swallowed up by some bacteria millions of years ago. So when we look at mitochondria and chloroplasts, we see the second membrane. suggesting that these structures were at one time swallowed up by another cell. Now that it seems a little far-fetched until you start thinking that, until you realize that each of these have their own DNA, separate from the cell's DNA. So each of your cells has a mitochondria that has a DNA that's different from the DNA that you have, that has the genes that make up who you are. And each of the chloroplasts in every one of the photosynthetic organisms on the planet, every one of the plants on the planet, every eukaryotic organism on the planet that does photosynthesis, there's DNA inside the chloroplast that's separate and different from the DNA that's in the plant that's doing the photosynthesis. So those two things are strong indicators that these were separate organisms and probably bacteria that were swallowed up through the process of phagocytosis. But then the question then becomes, is there any other evidence? And the answer is yes. The ribosomes that are inside these two organisms that are coded for by the DNA that's in these two organisms matches the kind of ribosome that you would find in bacteria. A lot of the genes that you find in these two organisms match the kind of genes that you find in bacteria. So there's all this evidence that strongly suggests that this idea of endosymbiosis is probably correct, and that these two things were at one time single-celled organisms, free-living, and that over time, after generation, they've lost the ability to live on their own. And of course, the cells that have them inside them, the plants in the case of the chloroplasts, and mitochondria, of course, in the case of the plants, and the animals and plants in case of mitochondria, can't live without these organelles either. So we have this symbiotic or close relationship that's commensalistic, right? So each side gains. Actually, it's not commensalistic. It's a mutualistic relationship. Remembering that commensalism is where two organisms have close relationships where one benefits and the other neither benefits nor is hurt. But in this case, these early organisms had a relationship where both benefited. In the case of the plant and the chloroplast bacteria, the plant got ATP, it got sugar from the chloroplast, where the chloroplast got movement, where it was able to be moved from one place to the other, and the larger cell was better able probably to gather resources necessary for the chloroplast to do photosynthesis. In the case of the mitochondria, the larger cell had more resources and was better able to get the mitochondrial bacteria what it needed to do, what it had to do. And of course, the mitochondria provided ATP for the cell. So that's a mutualistic relationship early on. No longer do we call it kind of a mutualistic relationship? It's no longer a mutualistic relationship because they're not separate organisms anymore. These structures have become one organism. They can't live without each other. They're one organism. So these are now considered organelles. But it's important that you understand this concept of endosymbiosis. It's important you understand that these three, this creature, the bacteria, this class of life that we call bacteria or prokaryotes, right, have a lot of similarity between mitochondria and chloroplasts. And today in this lecture, I'd like to focus on this chloroplast and the reactions it does. Okay, so the whole objective here is to look at photosynthesis as it's happening inside a chloroplast. And we want to make those connections between this process that's happening here in the chloroplast and respiration that's happening in the mitochondria. We talked a little bit about that a moment ago, about the similarity between mitochondria and chloroplast. The process is similar, and I think you'll see the same players that you saw in respiration come out here in photosynthesis. A lot of the processes are the same. Of course, some are working backwards. For instance, in the Krebs cycle, we were taking and making CO2 and getting NADH out of it, where here in the Calvin cycle, we're using NADPH and ATP, and we're taking CO2 and we're making this glucose molecule. We started out in the Krebs cycle. We started out with three carbon pyruvate. Here we're ending up with a 3-carbon glyceraldehyde 3-phosphate, and that ends up being turned into glucose in the end. These are light-dependent reactions, and they happen in the stroma, or light-independent reactions, excuse me, also known as the dark reactions. That does not mean they don't happen in the light. That means they don't need light to occur. So the Calvin cycle, or the dark reactions, they happen. They're taking the CO2 that's pulled in from the stoma. Remember, the CO2 is coming in through that open pore that's in the leaf, on the other side of the leaf, and it's being taken into the chloroplast. And this enzyme, this really important, super important enzyme we'll talk about more later at the end of the year, this Rubisco enzyme takes CO2 and makes it into glucose. Ultimately, it makes it into glucose. Now, it uses NADPH. Now, in respiration, we talked about NADH and FADH2. Here in the chloroplast, we're dealing with NADPH. A little different, but still, the whole point is it's carrying electrons. And ATP. So we need to put this energy, the energy in the electrons, the energy here in ATP, and put it into this molecule that we call glucose. Now here in this photosystem, this is called Photosystem 2, and this one's Photosystem 1. We'll look a little more in detail on that, but I don't want you to sweat it too much. What these systems do is they take the energy from light and excite electrons. The very first reaction is that they take water. In the whole process, their very first reaction, Photosystem 2, takes water, breaks it up into oxygen and hydrogen ions, and creates a hydrogen ion concentration just like the electron transport chain over in the mitochondria. And Photosystem 2 then excites the electrons, creates NADPH, creates ATP. Again, as it goes on to Photosystem 1, the same thing happens. Really, you're making a lot of ATP through that chemi-osmotic gradient that we talked about in the electron transport chain, using ATP synthetase just like there is in cellular respiration. But you need to create that hydrogen ion concentration, and that hydrogen ion concentration on one side of a membrane is happening through the excitation of electrons using sunlight. So again, what are the main points here before we go on? We're talking about glucose as being the molecule, okay, the product of the dark reactions, and here it is, glucose. And the whole point of the process, the whole point of having this chloroplast at all, is to make this molecule, to put the energy into glucose from ATP. We need to make ATP. How are we going to make it? We're going to use a chemi-osmotic gradient. So we have to remember these same terms we used before. Chemi-osmotic gradient. All right? It's not, remember, don't panic. All that means is you're going to use from high to low. We're using high to low. Okay? We're moving the hydrogen ions from one side of the membrane to the other, making this ATP. That energy is then transferred over to the glucose molecule. Then the Calvin cycle is using Rubisco, which is going to capture the CO2 and turn it into glucose. So in the light-dependent reactions, a photon of light excites the photosystem, the electrons in photosystem 2, breaks up water into oxygen and hydrogen ions. The hydrogen ions help create the hydrogen ion concentration difference in the chemiosmotic gradient that then drives the production of ATP, just like in the electron transport chain when we looked at ATP synthetase. So here we have ATP synthetase again, and that's going to produce this ATP. Okay, let's go on and look at it in a little more detail. I hope that by the end of this you'll look at it and maybe we'll watch some more videos. I'll send you some more support. Hopefully you've read the chapter already in class or at home in your textbook. Okay, when we look at the photosystems a little more closely, this is the light-dependent reactions. you can see that both of them, photosystem 2 and photosystem 1, the whole purpose is to excite the electrons. The electrons in water have very little energy right now. They're very stable. So what the chlorophyll does is it uses light energy to rip off the electrons and excite them, bring them to higher energy level. You learned about energy levels from Mr. Dissipate and from myself. And you know that these higher energy levels, the electrons are more unstable. They're able to do reactions much more quickly. And when you break up the water, just like you saw in electrolysis in the lab, when you did your experiment, you can take water and break it up into oxygen and hydrogen ions. Now, in this case, the electrons are taken away, so this does not become hydrogen gas. Instead, it becomes hydrogen ion, and at hydrogen, that's H plus, right? There's two of them for every water molecule. Those are creating that high to low concentration. So here's your high, here's your low. And you have these electrons that are going to be transported down the electron transport chain. As the electrons are transported down the electron transport chain, the electrons get less and less energy. ATP is made through that process. The energy from the electron, as the photon of light hit the chlorophyll, the electron was passed down the chain, allowing for hydrogen to cross the barrier to turn the ATP synthetase to make ATP. Now, to review, here's a question. Go ahead and answer it and put it in your notebook after you listen to this video again. The question really is, I think I said the answer like 3 times. Where is this ATP energy coming from? Where's the energy in ATP coming from? Both originally and then explain the process. So here you have the electron down here when it reaches photosystem 1. So it went through photosystem 2. The electrons got lost all their energy. All that energy was put in ATP. ATP is going to go over to the Calvin cycle. They're here at this very low energy state. Another light photon, another ray of light is going to hit this electron. It's going to excite it again. It's going to be a high energy electron. At that point, the high energy electrons, 2 of them, the 2 that we got from over here from water, when we broke water apart into oxygen and hydrogen ions, those same 2 electrons are then added to NADP plus. Now remember, NADP plus, when they take electrons, they also take hydrogens. We learned that from cellular respiration, from the Krebs cycle. When you get NADP plus, what happens is an H comes along and forms NADPH, just like NADH. You get 2 electrons and you get the H put on there, and this NADPH goes back over to the Calvin cycle, which we'll talk about next. Now here again is a question, where are these electrons from? Now there is a lot more detail known about this, about photosynthesis, and in fact we've learned recently that there might be some quantum forces involved in this, but really at this level, this is the process you need to know. You need to know where do the electrons come from, where in this whole process, Calvin cycle in which photosystem is water used, what is produced when water is used, why is water necessary, what is this whole process getting from the water. Where do the electrons get their energy? This is the question here, implied here. Where did ATP get its energy? Where is the electron transport chain used in photosynthesis? Where are the electrons that we got from water? Where are they going? Where'd they come from? How did they get excited to begin with, and how did they get excited again after they gave up all that energy? Those are the questions really that we're going to be looking at for the exam. Look here at photosystem 2, look at photosystem 1. This is really tricky that photosystem 2 actually happens before photosystem 1. All the processes of photosystem 2 actually happens after photosystem 1. Okay, let's go look now at the Calvin cycle. All right, so when we're talking about the Calvin cycle, we're talking about making this sugar, 3-carbon sugar, that ultimately will make glucose. It can make fatty acids and amino acids as well. This is why a plant can make its own food, and in fact does make its own food. We call it an autotroph. It does need other nutrients as well. It needs, obviously, here you see carbon fixation. Nowhere in here do you see nitrogen, which is part of the amino acids, right, that you need. Nowhere in here do you see a lot of the other components necessary to make the parts of the plant that it needs for functioning. So it gets those through its roots. So you have bacteria in the root nodules that are fixating nitrogen, etc. But this, when we're talking about the Calvin cycle or the dark reactions, we're talking about making glucose. We're talking about making sugar. But just always keep in mind that there are other organic compounds that you can make, assuming you have all the other components readily available. So in the chloroplasts, remember we talked about the light reactions just a moment ago. Water comes in, oxygen comes out, O2 comes out. The hydrogen from the water is used in the electron transport chain. Light's exciting the electrons. You're making ATP, you're making NADPH. Both of those are coming into the Calvin cycle. The very first thing, the most important enzyme, arguably the most important enzyme on the planet, the enzyme that literally changed the world, is this enzyme called Rubisco. We'll talk about it later in a little more detail. But Rubisco, the main purpose of Rubisco, the purpose for Rubisco is to fix carbon, to take carbon dioxide out of the atmosphere. When carbon dioxide is taken out, it's incorporated into these biomolecules that you see in the cycle. You see all these arrows because there are many turns, many different steps in the cycle. Remember, I'm not really interested in you knowing all these intermediate molecules. All I really need you to know is this enzyme for the Calvin cycle. All you really need to know is this enzyme called Rubisco, and that it fixes carbon. In other words, it takes carbon out of the air and puts it into this carbon chain, which ultimately gives you glyceraldehyde 3-phosphate, and that comes out and goes into sugar. The rest of the cycle goes back to produce this RUBP, this ribulose bisphosphate, and this is again the beginning of the cycle. It's also the end of the cycle. That's why we call it a cycle, because you end up with what you started with, minus this 3-carbon sugar. Now, if you look at it carefully, you're going to see that ATP is going to ADP, NADPH is going to NADP+, ATP is going to ADP. This is where we use the energy. Where's that energy going? That's the question. I said it before. I think I said it in the light reactions. I said it again. I'll say it again later. But I want you to think about it and try to answer that question. Why is this ATP going? Why are we using all this ATP? Well, not we, but the plants. Why are the chloroplasts using all this ATP and the energy in NADPH, all those electrons, all the energy in those electrons? Where's it all going? And how is that molecule, if indeed it goes into a molecule, how does that molecule act and react in the plant and in the ecology in general? Okay, so just a quick summary. Rubisco is the most important enzyme you'll ever hear of. It changed the planet. Its main purpose in the Calvin cycle is to fix the carbon, to take the carbon dioxide out of the air and put it into a molecule. There's a question that you have to answer for Tuesday. Where's the ATP going? Where's the energy in these 3 high energy molecules, ATP, and NADPH? Where is that going? And as further summary, I want you to remember that the light reactions, the original, you had the light coming in, exciting the electrons, breaking water, making oxygen, that ATP and NADPH, that energy that was in these came from the sunlight. Now the question is, why are we using these up? And one more question, how many of these CO2s do you need to make this G3P? Think about it. The answer is obvious if you can think about it. You have how many of these CO2s do you need to make one G3P? How many turns of this cycle have to happen in order to make one of these G3Ps? So really, that's the question in front of you. So here's a mitochondria and chloroplast in a comparison chart, a Venn diagram. take a look at the similarities between the chloroplasts and the mitochondria. Look at both, where they're similar and where they're different, okay? Always remember that mitochondria are found in both plant and animal cells. Chloroplasts are found only in plant cells. Okay, so make sure you're taking a look at all these and knowing the differences, because this will definitely be tested along with the rest of photosynthesis. Okay, here we have a diagram. It's kind of a summary diagram. We've gone through the light reactions and the Calvin cycle. We'll have some more. We'll do some more work on this on Tuesday and Wednesday. Your test will probably be Thursday or Friday. So make sure you're going over respiration, cellular respiration. That is including glycolysis. Make sure you're going over fermentation, the Krebs cycle, the electron transport chain. As you can see here, the electron transport chain is going to come back again during photosynthesis. Also go over photosynthesis on your own. You've read the chapter in class. You've gotten a couple videos now that kind of put it all together, hopefully. We'll have more time on Tuesday and Wednesday of next week to go through this again. I'm about to review it one more time as a whole. But make sure you understand photosynthesis. What do you have to understand about photosynthesis? You have to understand the light-dependent reactions, where you're getting the sunlight and you're exciting the electrons. And you also have to understand the Calvin cycle, where we fix carbon, take carbon dioxide out of the air and make it into glucose. So when you're looking at the light-dependent reactions, you have to remember that we're starting with step one. We have this photosystem 2 that's taking the electrons away from water, making oxygen gas, which is leaving through the stoma. Hydrogen ions are going to be used up in this system. They're going to create the hydrogen ion gradient. You have the third step is electron transport chains. After the photon excites these electrons, you get the electron transport chain that allows for this electron then to give all its energy to ATP through ATP synthetase. We'll look at that again here in a minute down below. You get this photosystem 1 now. Again, another ray of light comes in. You notice these yellow bands of light. This another ray of light comes in, excites that electron. It goes down and enters NADPH, goes from NADP to NADPH, because that hydrogen ion has to come along with the electrons. That then goes to the Calvin cycle. So this ATP goes to the Calvin cycle, this NADPH goes to the Calvin cycle. So let's take a look at 1, and then let's take a look at 2, okay? So 1, we're getting the hydrogen. We're using the electron transport chain, you see it here, to pump the hydrogens into the thylakoid space. The thylakoid space is inside the thylakoid. The interior of the thylakoid has a high concentration of hydrogen ion, very much like the intermembrane space of the mitochondria. And here, just like in the mitochondria, you have an ATP synthesizing enzyme. All right, so you have this hydrogen high concentration. It moves through the low, from high to low, turning the ATP synthetase, which then makes ATP. See, here it is. So the first step is to create the hydrogen ion concentration, the gradient. That's photosystem 2. That's what we're doing. And here is where we're making the ATP that comes here. It goes from ADP to ATP. There it is. You can see it happening. Hydrogen ions here, high, low, moves through, ADP goes to ATP. Again, that ATP goes to the dark reactions in the Calvin cycle. We call them light-independent reactions or dark reactions, because they don't need light to happen, but they happen in the day as well. They're independent of light. Okay, that's kind of a summary. I hope all these graphics help. Again, we'll go over it, and we will be printing these on Tuesday, but if you're not going over it this weekend, if you're not going over cellular respiration, again, glycolysis, fermentation, Krebs cycle, electron transport chain, all the steps, what's being produced, where's oxygen being used, where's water being produced, what is happening to that glucose molecule, where's glucose broken up, what are the 2 steps in glycolysis, when does fermentation happen versus the Krebs cycle and oxidative phosphorylation, or what we call the electron transport chain. When does one happen versus the other? Why does the Krebs cycle? What is the purpose of the Krebs cycle? What is the purpose of the electron transporters? Here again in photosynthesis, the similar questions are going to be asked. Where is the light being used? Where is water being used in the process? What's NADPH doing? What's the ATP doing? What is photosystem 2 doing versus photosystem 1? Here you see photosystem 2 getting the hydrogen ions after it breaks up water, producing this hydrogen ion concentration. Where is ATP being made here? These are the questions that are going to, what's the purpose of, what's the end product of all this? Glucose. How many carbon dioxides do you need to make one G3P? Or how many? Which remember if you remember correctly, that's glyceraldehyde 3-phosphate. Rewind to look at that molecule being made. How many glyceraldehyde 3-phosphates do you need to make one glucose molecule? It's really simple addition. I don't want to make it sound harder than it is. I want you to think, though. I don't want to tell you what it is. I want you to think. They call this a C3 cycle. Why do they call it the C3 cycle? Okay, so think about these things. The big questions and the overall idea is what I want to know. There's only one enzyme in this whole there may be two enzymes maybe in this whole process you need to know. Well, you got the photosystems, right? Which is a whole group of enzymes in different structural molecules. You need to know ATP synthetase, which you already know from electron transport chain, and you need to know Rubisco from the Calvin cycle. Really, the only one enzymes you need to know. You need to know the electron transporters. That's NADPH. It's NADPH. You need to know where ATP is being made and where's that ATP going. And then you have to answer the questions I've asked already in the video. The big one: where's the energy from ATP when you make ATP go to ADP here? When you produce NADP plus here, where'd that energy go? Where did that energy go? You should know it. Again, the answers to my questions are pretty easy. They're not a trick question. All right. Great. I'll see you on Tuesday. Email me any questions. I'll try to shoot you some more videos giving you some support. I will correct any issues with missing work on Tuesday. So make sure you have everything ready to go. And try to have a relaxing weekend even though you got all this work. All right. Bye-bye.